Welcome back, Mindsetters. Welcome back, Matrix. I know all of you have been waiting for this moment. When is grade 12? Well, here we are. And today we are chatting about DNA, the code of life. And I'm sure Kathy is going to take us through everything. Guys, please don't forget that I'm sitting here behind my computer. I'm not just sitting here chatting to Kathy and making jokes. I'm actually chatting to you guys. I am working. Believe it or not, this is a job. It's a pretty cool job, if I must say so myself.
Yeah, but it's also, you need to people, sorry, Indy.
No.
You've got to let Indy know whether you understand or you don't. Remember,
Totally.
I can't see you. And it's a difference between this look versus, that look. I can't see your faces. And I'm not talking to you, so I can't hear you. You can sometimes pick it up in someone's voice. Whoa, hold on. Don't understand. Now the advantage is that Indy's never done life science. So if I know if she understands, then I know that a lot of you, I've got through to a lot of you out there. She's our contact. She's like our
in-betweener.
Yeah, our double. Yeah. No, is it a double ganger? But
I know what you mean.
She's sort of like the link between you and me. She's going to say, hold on. Sophiso says this. James says that. Mary says, no. Please explain again. You need to communicate with her, and it's quick. I mean, it's sure. You know how fast the Facebook thing is. But I'm going to start, and I'm going to go through it, and if there's a problem, please, Indy.
Yeah. Facebook.com/learn extra. Chat us on our page or chat us on our events wall. You'll have a look and you'll see in our events we've got a Mindset Learn Extra Life Sciences every Wednesday show. You can ask your questions in there. Also, guys, help each other out. If one of you know the answers and you can see on the wall that somebody said, I don't understand, please don't be afraid. Write to them, chat to them, guys. Let's get this. We can all start talking.
That's the best way to actually learn, I hate to tell you, is to sit and teach someone else. And when you're explaining to someone else, you actually have to get your facts straight. And what you're doing is you're reinforcing what you know.
Yeah.
So people help each other. And we'll try and help you as much as we can too. But please, if there's anything you don't understand, anything you want to ask, anything you're confused about, anything you want me to repeat, tell Indy. All right. So. Cool. Can I start?
You can start now, Kathy.
All right. What we're doing now, people, DNA, code of life. So what on earth are we talking about here? We're saying DNA, code of life. What is this? This is all fancy words. No, it's not. Everything you are, from the little dimples, from your crooked pinkies, from the shape of your mouth, the shape of your ears, your fingerprints, whether you've got big feet, small feet, round feet, bunions, all of those, everything about you is written as a recipe in your DNA. Now, remember, DNA was found in the nucleus, and the nucleus had the chromatin network, and which was just all your DNA in a mush. All right, now, your DNA has the answer to who and what you are, because your DNA makes up your chromosomes. Okay, so I'm going to move through my slides here, and the first thing we have to look at is part of DNA and what DNA is, and that's a deoxyribose nucleic acid. We also have a ribose nucleic acid, and all these words, and you're thinking, whoa, I have no idea where all these words come from. Look at the beginning of a word and the end of a word. Okay, so you look at the first part of the word or the last part of the word, and it will tell you what the word is about. Okay, so if we look at nucleotide, a nucleo would be part of a nucleus. So before we do anything here, we need to look at, and I need a blank screen, which is what I was trying to get to. We look at a nucleus. And inside that nucleus, what am I doing? Let me start again. All right, there we have a nucleus. And in that nucleus, we are going to have chromatin network. Which is like just a ball of mush. And that chromatin network, the word chromatin comes from chromosome. So what we're going to have in there are our chromosomes. And chromosomes, remember, people are always found in pairs. We always have, like socks, you have a pair. So you have 2 complementary chromosomes. One you got from your dad, and one you will have got from your mom. And if you were a peach, if you were a kitty cat, if you were a locust or a fly or whatever thing, organism, living organism you want to think about, it would have a set of chromosomes. One set that it got from its male parent and one that it got from its female parent. All right, so you put the 2 together and that becomes the offspring. Now, looking at the whole process, you've now got chromosomes. Sitting on the chromosome, which is 2 chromatids joined by a centromere, you're going to have your DNA. Now, DNA and RNA are 2 types of nucleic acids. So we look at our nucleic acids, and those nucleic acids, we have DNA and we have RNA. And the DNA and the RNA is basically based on whether there are oxygens and the sugars that are available. So DNA is deoxyribose nucleic acids. Okay, deoxyribose, and ribose nucleic acid is RNA. Right, now RNA is one strand. It's a single little strand, and it's basically there as a helper. So we have messenger RNA, we have transfer RNA, we have ribosomal RNA, and the RNA dots in and out in little pieces of 3 or little chains of 3, bits and pieces, and it sort of floats around the cytoplasm and in the nucleus and in around the nucleus and out again into the cytoplasm. So the RNA is an uncomplicated structure, and your DNA is a double strand. So you've got these 2 strands that bond together with weak hydrogen bonds and twist. So it forms what we call a double helix. Okay, so those are our nucleic acids. What does a nucleic acid consist of? Well, it consists of nucleotides. So if we look at a nucleotide, immediately gives you the clue that it's from the nucleus. Okay, so we have a nucleotide. One nucleotide is a basic building block. Now, if you watch the grade 10s, and if there are grade 10s watching today and now, part of the grade 10 syllabus is the whole chemistry of life, the beginning of the whole process, the basic nutrients and structures and organic compounds we have in our bodies. So one of the organic compounds, it can only be made by a living structure, and it has to contain carbon, which it does. Okay, is your nucleotides. So what have we got? We've got our phosphate ion, which is an element. Okay, we have our pentose sugar, which is a compound. Why? Because this pentose sugar has 5 carbons. That's why penthouse has 5 rooms. Pentose sugar it's a 5 carbon sugar. Sugar. Okay, pentose sugar and nitrogenous base. This is my protein part. And all your DNA and your RNA is, we say it's part of protein. It's made of protein, and it's the protein there. It's your nitrogenous base. That's your protein part. So without proteins, we can't make nucleuses or nuclei, because we can't make chromatin network, because we can't make DNA. which would mean the person would die because they would not be able to replace their cells. Because all your recipes are written in the nucleus. Now, your nucleotide, this is the basic building block of DNA and RNA. In RNA, you're just going to have one big stripe of phosphate, pentose sugar, nitrogenous base. And then your next phosphate will join here, pentose sugar, nitrogenous base. And you have just this long chain. And we say that the phosphate and the pentose sugar, the two together form the backbone. And the nitrogenous base, or if you can't spell it, nitrogenous, means part of a nitrogen. Nitrogenous base is what links the two, it sticks out. So when we have DNA, we're going to have a double strand. RNA, one strand. Okay? So now, very important, and this is something that many learners battle with. And I promise you now, stop doing whatever you're doing. Stop taking sips of anything. Stop listening to your little sister or brother playing on the ground. Don't listen to the cars outside. Don't listen to your mother or whoever is busy with supper. I want you to close your brain and focus purely on what I'm going to tell you now. Because if you understand A, you will be able to remember. And if you remember this, and you understand it, I promise you now, on my absolute word of honor, you will get full marks for DNA replication and protein synthesis from now until you have children, and you'll be able to teach it to them, and you'll be able to teach it to your grandchildren. So I want you to focus completely on what I'm going to do here now. Okay, we have, just to recap, we have nucleic acids. They are acids that we are going to find in the nucleus. Okay? Nucleic acids are organic compounds. Okay? And they are made up of DNA and RNA. DNA is a deoxyribose, double-stranded, twisted structure, contains all your genes. Not the things that you wear, blue jeans, I'm talking genes, what you are. The code of life. And RNA is there to assist. And the RNA can go anywhere it wants to, where the DNA is only found on the chromosomes in the nucleus. Okay, now, the DNA and the RNA is made up of basic building blocks. Those basic building blocks, the basic building block is called a nucleotide. The nucleotides always have a phosphate ion, and it's not iron like the thing that you iron clothes with, it's an ion. A pentose sugar, a 5-carbon sugar, pentose means 5, and a nitrogenous base. This is the protein part. That's why we say proteins are so important, because if you don't have proteins, you can't make DNA. And if you don't have DNA, well, guess what? You're dead. Okay, now, and I must stop saying okay. I think it's the 5th time I've said it. So
okay.
I'm trying to focus very hard here and get to this right. Now, here comes the really, really important part. I need you to get this in your heads. First of all, nitrogenous bases. So in our nucleotide, we have a phosphate ion, pentose sugar, phosphate ion, pentose sugar. So that forms the backbone of the RNA or the DNA. Phosphate, pentose sugar, nitrogenous base. And the nitrogenous bases are divided into 2 groups. Your first group are your purines. They're pure, purines. And then your second type or group of nitrogenous base, those are your pyrimidines. So you have your purines and your pyrimidines. But now it gets one step, a little bit more complicated. We say, okay, now the purines are further divided into two different types. Number one, adenine and guanine. So nitrogenous bases, which are part of your nucleotide, they attach to the pentose sugar. We have purines and pyrimidines. Purines is a nice short little word. Pyrimidines, whoa, what has pyrimidines got that we can remember? It has a Y. Right, so purines, pyrimidines. Purines are divided into two further groups. So they're subdivided. And we have adenine, so adenine if you want to call it that, or guanine. So adenine and guanine. In other words, let me do it like this. Instead of going A, B, I'm going to say A for adenine and G for guanine, which you can't see because this is red. Right, so A and G. Those make up your purines. You have to know these words, people. You've got to learn them. So, nitrogenous bases, purines, and that's divided into adenine and guanine. Okay, now, we look at our pyrimidines, and whoa, hold on a minute. Pyrimidines has a Y in the word. And look how clever this is. Pyrimidines, the two, is cytosine, which has a Y, and thymine, which I'm coloring green on green, which is rather silly, but pyrimidine, thymine. So the pyrimidines, the word has a Y in it. That's how you're going to remember it. And cytosine and thymine. But that's in DNA. But in RNA, well, you've got to have something there as well. In RNA, instead of thymine, it's got a slightly different structure, so it's got a different name, and that is uracil. But the difference between a U and a Y is just a tail, really. So you've got pyrimidines are cytosine and thymine, but in RNA, because it's only one strand, whatever excuse you want to use, it only has uracil, not thymine. So if we look at this and we say, all right, we're going to say adenine and guanine. Now you know that those are part of your purines. They are going to always join because they form complementary nitrogenous bases. They are complementary. Now, if you find, girls and boys, if you find the guys, if you find a perfect girl out there, she is beautiful. To you anyway. She is bright. She is intelligent. She likes you. She listens to you. Blah, 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 blah. Okay? And she is perfect. And you think the two of you complement each other. The things that she's strong in, you weak in. The things you strong in, she's weak in. You help each other, you guide each other, and the violins start playing. You are complementary. Now, with DNA, exactly the same. Because remember, it's two strands. So you're going to have two lots of nucleotides bonding together. So, your purines, okay, adenine and guanine. All right, now I'm going to use another color here. Let's use blue. And we say, on the other side, we're going to have thymine, because we're looking at DNA. Thymine and cytosine. So it's thymine and cytosine. And you're going to battle to see the blue, and I'm not sure. Okay, well, let's go. All right, so we got thymine, and it has the Y in it. And we have cytosine, and it's got the Y in. So immediately we know it is purines and the pyrimidines. All right, the Y, and the Y. Okay, except if it's RNA, we're going to have uracil instead of thymine. Now, watch. We're going to take these complementary bases and we're going to put them together to make, so that all of this makes DNA, which is a nucleotide bonded to another nucleotide on the other side. And this bond is a weak, now listen carefully, it's a weak hydrogen bond. And it breaks very quickly, okay? So, there we go. Our complementary bases, adenine, which is a purine, will always bond with thymine. And guanine will always bond with cytosine. So think of this, it's AT and GC. And when you have RNA, well, hello, RNA doesn't bond to anything. RNA is a single strand. So instead of thymine, what do we have in RNA? We're going to have a uracil with a U. And with this, I'm going to leave you. We're going to go for a quick ad break. I want you to close your eyes and try and picture what I've just told you now. Adenine to thymine, cytosine to guanine. And those are the only combinations you're ever going to have. So close your eyes and make sure you remember it, and I will see you in a minute.
Welcome back, guys, and I hope you're really enjoying the session. Grade 12s, where are you on the page? There's not that many of you. I want to do a shout out to Tumelo, Skills, Hudson, Siabonga, Tatonero, and Anza. You guys are rocking the page, and I'm loving chatting to you. And thanks for all your amazing comments, and thanks for the questions. Keep them coming in. I know a lot of you are asking one particular question, and Kathy is going to set the record straight. Okay, Kathy, take it away.
Okay, I've just heard protein synthesis, DNA replication, protein. I cannot do that with you until I know that you understand this. So, purines, pyrimidines. Adenine will always, only ever on this planet bond with thymine. So it will always be A to T, at. And guanine and cytosine will always bond together. Doesn't matter whether it's on this side or swapped around. Adenine and cytosine are always together, and guanine and cytosine are always together, and adenine and thymine. Purine, pyrimidine. People, get that in your heads. If you've got this, I promise you now the rest is a piece of cake. So now we go down, and let me go down, 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 down. I want to show you this pretty little picture, and what it does is it shows you, there you go, adenine and thymine, and they bond together. There's your weak hydrogen bond, and you know you've got this, so that should be adenine, that should be adenine, that should be thymine. It tells you the opposite. Now remember, if this is adenine, that has to be thymine. So if we know that that is adenine, this one must be thymine. Why? A to T, G to C, C to G, T to A, ever, only, and that's the way it is. All right, now let's continue. This comparison, what we'll do is we'll put this on the website as well. It's there, it's in the X sheets. Have a look. You must know this difference. They love asking it. They'll say, name 2 differences, name 3 differences, tabulate one difference between, make sure that you know it. It's easy learning. And it's honestly not hard to remember either if you just use your common sense and you think of the basic structures. All right, and that was it. And I think I've just told everything. Now, I'm going to just give myself a clean screen and we go from here. Right, now listen carefully. DNA replication. If I want to replicate something, I need to make the same. All right, so let's say I want to replicate pages for my 5 friends. I've made wonderful summaries, and I want to make copies for my friends. Then we say it is DNA replication. Now, when would DNA replication occur? Now, use your common sense. When would it happen? Maybe when I want something that's identical, so possibly doing mitosis. Yes. What do I want to do? I want to make something that is an exact copy. But hold on, in meiosis, which is for sexual reproduction, I must make sure that the DNA is perfect for when meiosis, which is the halving of the nucleus, and the halving of the chromosomes, the chromosome pairs, when they separate out, so you've got your pair, one chromosome will go this way, one goes that way. We need to know that when those when the DNA was duplicated, it was perfect. So that is DNA replication, incredibly important. So now what happens? Well, we're going to make an exact copy. So first thing, we're going to have our strand of DNA, okay? And we're going to have our phosphate. Okay, this is going to be step 1. Then we're going to do step 2 over here. So we have our strand, so phosphate, we have our pentose sugar, phosphate, pentose sugar, 5-sided sugar, phosphate, 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 pentose sugar. Okay, I'm really trying to do this quickly. So here we're going to have our nitrogenous bases and our nitrogenous bases so that you've got some idea. But it looks like, if I take that and I simplify it, it's going to look like this. It looks like a ladder or a zip. Okay? We've got our nitrogenous bases here in the middle. And remember, there are always 2. It'll be a purine plus a pyrimidine. And it is always adenine bonds to thymine and cytosine bonds to guanine. Always. Okay. So I want to copy my DNA. So what does it do? It says, right, hold on. Easy. Step number 1. There's an enzyme that's going to break those hydrogen bonds. Now, I really need to get myself a color here. So, it's going to break these hydrogen bonds. That enzyme comes along and it breaks them. But as it's breaking them, so we are going to end up with, and I'm going to do it in the opposite direction, we're going to end up with, this is point 2, this happening. That's that one there. And then this one. And then my nitrogenous bases. So that's this one here. And that one. Okay. So the enzymes cause those weak hydrogen bonds. Let me write nicely here. Weak hydrogen bonds. And they're going to break. Step 2 is it opens up, it unzips. Okay, happy. How are the responses there?
Yeah, very, very happy.
Okay.
They actually think that you're a fantastic teacher.
But
I'll read it all the nice
and then I will
after.
afterwards say, whoa, you're so sweet and nice. And you make me feel good. But this is my job, is to teach you. So pay attention, okay? Remember, adenine to thymine, cytosine to guanine. All right, so step 1, we have our process. Step 2, we are now going to have the weak hydrogen bonds breaking and an unzipping. So this here would be, I'm so worried about the colors that are clashing here. But okay, if we look at this, we say this is strand 1. And this will be strand 2. All right, people, just bear with me for a second here because they can't see the colors. So let's make this, let's try
bright green.
Yeah, let's try Lumo green.
Yeah.
Okay, let's see if this works. All right, so this is, as I've said, I'm going to just put STR1, and this is strand 2. All right, we've got the 2 strands, 1, 2. and those weak hydrogen bonds are breaking. So why on earth would we do this? Well, easy. This is what we want to duplicate it. So we say, okay, fine. Step 3. What have we got? We now have okay, you remember this? Right? There's my strand 1. There's my strand 2. And this is from the original DNA. So we'll do this. The white color is the original. So what's Kathy going to be doing now? I'm going to duplicate it. I'm going to what we call replicate. Now, if you replicate art, you're making a copy of the original masterpiece. Okay, and sometimes those copies are brilliant. Ours is going to be brilliant. It has to be perfect. So we say, right, no, look at this. We're going to use our lime green here. We say, along comes, so what was RNA's function? RNA's function was to say, hold on, in little groups of 3, they come and they slot in here. So let's do this. Let's just go back to that and we say, right, what have we got here? Let's say this is adenine, and this is guanine, and this is thymine. We're just going to work on those 3. So we've got AGD. So that's my original one, which means the complementary bases on strand 2. Oh, please tell me you're going to get this. Please tell me you're going to get this. Adenine 2, thymine, guanine 2, cytosine, adenine 2, thymine 2, adenine. Remember, people, A to T and G to C.
Can I jump in?
Yes.
We've got Sevi Piri. I hope now that I've said your name, you're happier. He said, he's given quite a nice little thing here. He said, he's understanding everything, and an easy way to remember AT is always together, and GT is good couple. What do you think of that?
AT is
always together, and
GC good couple.
Good couple.
Brilliant. Absolutely brilliant. So we've got good couple and always together. And if it's a bad couple, it's when you don't put A to T and G to C. All right. Now, and I promise you're going to thank me when we start with protein synthesis, because you're going to understand this. Right. So now, I hope my producer can actually see the bright green. Okay, so this is my RNA. And in that RNA, I'm going to have thymine. Am I? But let's do that. Thymine and adenine to cytosine, and thymine to adenine. So it's the complementary bases. But remember, this is RNA. And do we have thymine in RNA? No, we don't. Remember, it's a pyrimidine. But instead of thymine, RNA will always have uracil. So we are going to take this one away, so we never ever see it again, which is just done. Okay, and we are going to put uracil. Oh, why is it doing this? Uracil. Okay, people are going to have to be patient with me here, because I have to get used to this pen and things move around on the screen. And okay, so there I've got my RNA in a group of 3, and that group of 3, okay, is called a triplet base, okay, or a codon. Now, on this side, let's just do this. We had thymine there. We're going to have exactly the same process occurring. We're going to have, let's use the purple. We're going to have our RNA come in here, and thymine, but remember, no thymine. Why? It is RNA. So we take our thymine out. That becomes uracil and guanine and adenine. what have I done now? This strand has got 1/2, which is this DNA, at least this will become DNA. This will have 1/2 of the original strand. This one here is going to have 1/2 of the original strand plus that one. So when these two go apart, I end up with exactly the same DNA structures, except that step 4, I'm going to have, and this one will have the lime green, and this one is going to have the pink. But they are going to be identical. Why? Because always together and good couple. All right. Got it, people? Happy with that? So this would be one complete, because all the little rungs will be there. And these little rungs will be there. And I now sit with my DNA 1, which is my original DNA, and this is DNA 2. All right? Why? Original, original, duplicate, duplicate, but remember, you cannot mess up. Why? Adenine to thymine, cytosine to guanine. And this, people, is what DNA replication is, so that you end up with exactly the same DNA, a duplicate of the original DNA. But we have our 2, and each strand will always, each DNA will always have half the original one. Okay, so always half the original one plus half, which will be the new. And together, we'll end up with 2 brand new strands. Right, that is DNA replication. Most important thing you must remember is your purines plus your pyrimidines. Okay? Purines, adenine to thymine, cytosine to guanine. Always together, good couple.
Thanks, Sevi, for that.
Perfect. Okay,
Yeah.
so I think this has gone very nicely. I'm so chuffed.
I have 2 questions. You know when we said that if there is anything they're not understanding, they can re-ask?
Yes,
so I'm going to re-ask.
No,
okay.
they must.
Okay, so
I'm just going to have a sip of water though.
No, go. Well, I'm asking.
Okay.
Kazi Chorny wants to know, why do the DNA strands split again?
Okay, first of all, they're always going to be hormones. And before a process like mitosis, all right, you want to make a new cell. If you're going to make a new cell, you're going to make sure that the same DNA is in this cell as in that cell. Do you not agree? So it's got to be identical. Part of it is the DNA in the nucleus. So you've got to make sure it is absolutely perfect. And that's why DNA replication will take place. All right. Remember, in meiosis, you're halving the chromosomes. Here, we are duplicating because your DNA sits on the chromosomes. It's because of your DNA that the chromosomes are what they are, that they carry the information, the hereditary characteristics. Remember, this entire process here, if we look at the original heading that we had, and it's not going up, let's just see here. And this has just gone standstill. Okay, so it's on a mission of its own. But this is the code of life. It is everything you are is written in your DNA. Your DNA make up your chromosomes, and your chromosomes make up your nucleus. So every process that happens is originated there. Your body tells it, or the nucleus controls your cells. And hello, if it controls your cells, it controls the rest of you. Because cells make up tissues, make up organs, make up systems, make the organism. And that you know from grade 11. Okay? Any other questions?
Now I've got lots of other questions. Do you want to go to a break so we can unfreeze the screen, or are you cool to carry on?
No, I'm just going to just create my own new screen.
Yes, that's what I thought. I thought Kathy just likes to dance. Okay, so Nokatuli wants to know, she said, so Kathy, what is the old strand called again?
Nothing. Original strand, new strand. That's it. Remember, you're doing grade 12. You are not neurosurgeons. You are not doing a doctorate or a, I don't know, listen,
PhD.
You're not doing a PhD or a doctorate in genetics. The genetics and the DNA and code of life that you have to learn is look, I know it's hard for you, but it's actually very basic in the greater scheme of things. All you need to know is during DNA replication, step 1, okay, step 1, what happens? Those weak hydrogen bonds between the nitrogenous bases break. They unzip because of enzymes. And the DNA, the 2 strands of DNA will separate. They open up. Step 2, RNA comes along and the complementary bases line up. Step 3, the bonds reform, those weak hydrogen bonds reform between the complementary bases. Step 4, 2 identical strands of DNA, or 2 identical DNA molecules. The one strand A, or the first strand is the original plus a duplicate, and the other one has the second strand plus a duplicate. So they end up being identical. Okay? Happy?
Perfect, yeah. I hope so.
Any other comments coming in?
There are. I think that we could actually probably carry on until the end of the show. There's quite a few.
Okay.
But a lot of people are saying, like even Donham, I think that's how I say her name. Okay, thank you, he understands. But let's have a look. Okay. Dear Kasha wants to know, I don't seem to understand how the uracil joined to the adenine. I'm not too sure if I missed something.
Okay, listen. All you're going to have is RNA doesn't have the ability to have a molecule like thymine. So it's made a version for RNA which would be uracil. Okay, that's all you have to remember. So in DNA, okay, you are going to have your purines, you're going to have guanine and adenine, and your pyrimidines for DNA are thymine, and I'm going to put the Y here. And I'm going to put in brackets, the thymine and cytosine. All right. And when we look at RNA, we're stuck with this bright green, by the way, because this screen is definitely a male screen. Okay, RNA, your purines are going to be guanine and adenine, just like normal, and your pyrimidines are going to be cytosine, because it's happy with the cytosine, but instead of thymine, we have uracil, and that's it. And that is going to be how you are going to be able to tell in any exam or test that, oh, hello, there is a U there in that little diagram, that picture, that set of structures. Therefore, it is RNA. And oh boy, there's a T for thymine, but no U. It is automatically DNA. Even if I gave you half a strand of DNA, if there was a thymine there, then you know, it's DNA. And if there's a U, it's RNA. And that's how you're going to tell it. Right, so that's what you need to know. And by the way, this is another difference between RNA and DNA. RNA single strand, DNA double strand. RNA, it's a single strand that's untwisted, can't be twisted, it just stays. Whereas your RNA, double helix. There are hydrogen bonds here because there's a double. There are no hydrogen bonds. Thymine, uracil. Okay.
Cool. Okay, you ready for the next question?
Mm-hmm.
Okay. Bule Shabalala wanted to know, does DNA join with RNA for DNA replication to occur?
Does
DNA join with RNA for DNA replication to occur?
Yes, because the little pieces that float around inside the nucleoplasm, which is the cytoplasm that's inside the nucleus, okay, those little bits and pieces that float around, they are RNA. Remember I said your RNA can go anywhere. It can do whatever it wants to. So the RNA just comes along and says, hold on, hang on, we've been called. DNA replication taking place, guys. We need to slot in there and make sure that that original strand of DNA has its complementary bases and your little RNAs come in there nicely. So if that's my original strand, so my O1 and my O2, those are my original strands. There are my nitrogenous bases. Okay, choose A, T, G, or C.
C.
C. Next one.
G.
Next one.
A.
Next one.
Can I do another A?
Okay, there we go. All right. This side, what am I going to have people? Come, tell me. C, remember, C to G and A to T. And any question on DNA, code of life, in an exam, in a test, questions your teacher's given you, please, when you start at the top of your page, you write A equals T, G equals C, and don't think it's babyish. It's going to remind you every time. Even if you begin to get confused, it's going to remind you. So, what do we have? C to G, and G to C, and A to T, and A to T. Ha! T's are there. It is DNA. Okay, now we slot in. Now remember, I don't have a different color. So along comes, your RNA normally floats around in little threes, little groups of three. So it says, ching, 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 and it slots in there, another one slots in there, another one slots in here, and so it happens. So instead of cytosine, this one's going to have a G, here I'm going to have a C, and here I'm going to have a U. Same thing here. Here I'm going to sort of a G, I'm going to have a C, and here I'm going to have a, oh, stop it.
Is it giving you problems again?
Okay, here I'm going to have my G, and instead of a T, I'm going to have an A, and everybody's happy. So, and this keeps happening. Your little RNAs come in here, and they slot in, and they'll slot in again, and they keep slotting in until when we look again, we have 1, which will be O1, and N for new 1. And here we'll have O2 and new 2. And I now end up with my identical 2 DNA strands. Perfect.
So Tato wants to know, so does the new strand also contain pentose sugar?
People, the pentose, you're going to have pentose sugar every time. Remember, a pentose sugar is a 5-carbon sugar. So whether it's ribose or deoxyribose, it's the carbons that make it a pentose sugar. And this goes back to the whole process of what the chemistry of life is. Remember, if it's got, and this is an easy way to remember it, if it's got 1 carbon, okay, or 2 carbons, or 3 carbons, or 4 carbons, or 5 carbons, that's what makes the different types of carbohydrates. But just a simple one, mono is always 1, di or bi is 2, tri is 3. Tet means 4. This is all Latin. Pent means 5. Hex means 6. Okay, so that is what we have. So we go mono, di or bi, tri, tet, pent, hex. So if you have a hexo sugar, it's going to have how many carbons? 6. Okay. If it's only got 5 carbons, it is a pentose sugar. If it's got 4, it's a tetrose sugar. 3, a triose sugar. You follow? And that's what we're trying to show you here. So don't worry about your DNA and your RNA will both have a pentose sugar. Why? The phosphate, the pentose sugar, and the nitrogenous base is what makes up a nucleotide. A nucleotide is the monomer of DNA and RNA, so of nucleic acids. Because you get 2 types of nucleic acids, DNA, double strand, and RNA, single strand.
Okay, our producer is going to kill us. Okay, we've got 20 seconds. We've got 2 more questions.
Quickly,
really quickly. Where do the
Goodbye, but listen now for the next 10 seconds. Yes.
Where do the nucleotides that form a new strand come from?
They're floating around within the nucleoplasm, which is the cytoplasm inside the nucleus.
Okay.
And in and out through those nuclear pores.
And is DNA replication the same in animals and plants?
Yes.
Okay.
Any living organism, DNA replication has to take place if there is DNA.
Okay, last question. Discovery of DNA, will it be tested important?
Yes.
Okay.
Because they're going to give you one of those inane questions, Watson-Crick model, blah, blah, blah. So just make sure you know. Just read it. It's a PowerPoint of 4 slides. People, go back and read it. Nothing to kill you with. Have a wonderful day. We're done.
We did it.
Guys, good luck. Have a wonderful week. We will see you next Wednesday, same time, same place.
See you. Bye.
Bye.